Hello, everyone, and thank you for joining us for today's live webinar, ELISA-like Assay Formats and the Benefits of Assay Automation. I'm Christy Jewell of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. At Beckman Coulter Life Sciences, we are dedicated to improving the health of people around the world. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. For more information, please visit beckman.com. Now let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click Send. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I would now like to introduce today's speaker, Philip N. Gorman, scientist in antibody generation for biotherapeutics discovery at Boehringer Ingelheim Pharmaceuticals. Philip earned a master's degree in biology at Southern Connecticut State University and subsequently worked at the Yale School of Medicine, Perkin Elmer, Thermo Electron, and Pfizer. The majority of his expertise is in immunoassay development and validation. He also has extensive experience in lab automation and integration. Philip now oversees Boehringer's high throughput antibody discovery team, whose principal duties are primary screening, cloning, and transient expression of therapeutic antibody candidates. Philip Gorman will now begin his presentation.
Thank you, and thank you for joining us all today. And I will be presenting today's webinar on ELISA-like Assay Formats and the Benefits of Assay Automation. As a brief overview of what we're going to be talking about today, I have a couple slides on my own personal background and background of my employer, Boehringer Ingelheim. We'll go through a recap of the standard ELISA formats that most scientists are familiar with, and then some, as I refer to them, ELISA-like formats that are derived from this overriding principle. One that we will focus on is electrochemiluminescence, and that is primarily put out by a company called Mesoscale Discovery, MSD. So we'll go through an overview of the MSD's technology. We'll touch on implementing lab automation in varying scope and scale. I have an example of one of our own platforms here at Boehringer, which is an automated MSD screening platform that we have named Skynet. We'll do an overview of different forms of scheduling software and how they can be used using Beckman Coulter's scheduler, SAMI, as an example. And we'll touch on the needs and requirements of assay validation in the realm of automated platforms, conclusions, and then I'll be glad to take any questions. Boehringer Ingelheim is a family-owned global corporation with multiple subsidiaries throughout the world. It obviously was founded in Ingelheim, Germany, given the name. Their current focus is human pharmaceuticals, animal health, and it is also one of the larger biotherapeutic contract manufacturing groups in the world. BI has over 50,000 employees worldwide and an R&D expenditure of over 3 billion euro. Currently, the Biotherapeutic R&D headquarters is located in Ridgefield, Connecticut, with research sites in Austria, Germany, and, as I said, multiple subsidiaries throughout the world. And animal health is centered in Athens, Georgia. As for myself, I received a bachelor's in Physiology and Neurobiology from the University of Connecticut in 1999. I received my master's degree in biology from Southern Connecticut State University in 2007. I've been in the pharma and biotech industry for a little over 16 years and have touched in discovery, research, development, sales, field applications. I've worked for Yale University, Perkin Elmer, Thermo Electron, and Pfizer prior to my current position here at BI. The majority of that experience is in immunoassay development, along with liquid handling, automation, integration, and assay transfer. I have been an employee of BI since 2011. ELISA is probably the first assay format that most people learn. The acronym ELISA stands for Enzyme-Linked Immunosorbent Assay. And these assays are very easy to develop and run. There are multiple formats allowing for a significant amount of flexibility in how they are set up. There are drawbacks to each of those formats. So we'll look at standard, the four formats that most people are aware of: the direct ELISA, indirect ELISA, sandwich ELISA, and the competition ELISA. And then I'll also bring up what I refer to as a hybrid ELISA, and then ELISA-like or ELISA-derived technologies. The first assay format that we'll touch on is the direct ELISA. Direct ELISA is much faster than other ELISA techniques, as fewer steps are required. The assay is also less prone to error since fewer reagents and steps are needed, although there are some disadvantages to this method. So an instance in which someone might be using a direct ELISA is looking at an activated cell type and did my cells secrete protein X and how much. In a direct ELISA, only a single conjugated antibody is used for antigen detection. So if you are trying to assay a cell culture supernatant, let's say for 8, your supernatant is coded onto the plate, but the coding is non-specific. Everything in that supernatant is coded onto your plate. You're going to probe that plate with a conjugated anti-IL-8 antibody. So the only thing you're going to detect is IL-8. Direct ELISAs are less flexible since the specific conjugated primary antibody is needed for each target protein that you want to analyze. And as there is no secondary antibody used, there is no signal amplification. This reduces assay sensitivity. The second assay format that we'll look at is indirect ELISA. The indirect ELISA has a high sensitivity since more than one labeled secondary antibody can bind to the primary antibody, which results in a signal amplification. It is more economical than a direct ELISA, as fewer labeled antibodies are needed. You only need one, usually anti-species IgG or IgM, to detect any of the primary antibodies within your assay format. Indirect ELISA delivers greater flexibility since different primary antibodies can be used. Among the disadvantages is a possible cross-reactivity of secondary antibodies to the adsorbed antigen, which could increase your background noise. Also, indirect ELISAs can take longer to run than direct ELISAs since additional incubation steps are needed. The indirect ELISA is usually most suitable for determining total antibody concentrations within a sample. sandwich ELISA is another one that most people are very familiar with. A sandwich ELISA requires the use of matched antibody pairs, a capture and a detection antibody. Each antibody must be specific to target antigen, but they cannot bind a similar epitope. There can be no overlap in the binding epitopes. So in this type of a format, one of the antibodies will be coated down onto the plate. Your sample will be with an additional incubation. Then your second matched antibody is applied, and then you're labeled secondary. The key advantage to a sandwich ELISA is a high sensitivity, and it can be 2 to 5 times more sensitive than a direct or an indirect ELISA. And it also delivers a high degree of specificity since 2 antibodies binding to the antigen are required to detect it. And it offers a high amount of flexibility with setting up your matched pairs of antibodies. These types of ELISAs are particularly suited to the analysis of complex samples, since antigen does not need to be purified prior to the assay, yet still delivers high sensitivity and high specificity. The final standard ELISA format is the competition ELISA or blocking ELISA. This is the most complex of the standard ELISA techniques. However, each of the above assay types can be adapted to a competitive format. The competitive or inhibition ELISA is predominantly used to measure the concentration of an antigen or an antibody in a sample by detecting interference in an expected signal output. Essentially, sample antigen or antibody competes with a reference material for binding to a limited amount of labeled antibody or antigen, respectively. The higher the sample antigen concentration, the weaker the output signal, indicating that the signal output inversely correlates to the amount of antigen in the sample. Known antigen would be used to coat a multi-well plate. Following standard blocking and washing steps, samples containing an unknown antigen are then added. Labeled detection antibody is then applied for detection using relevant substrates such as TMB or ABTS. If there is a high concentration of antigen in the sample, a significant reduction in signal output will be observed. In contrast, if there is very little antigen in the sample, there will be a small reduction in the signal output. The versatility of these assay formats have given rise to, we'll say, much more complex and much more technology-based assay formats that I refer to as ELISA-like formats, and you might be familiar with some of them. ELISpot is an enzyme-linked immunospot assay that measures frequency and is primarily used for cytokine-secreting cells, but can get down to the single-cell level. For this particular type of assay, cells are cultured on a surface coated with a specific capture antibody in the presence or absence of stimuli. Proteins, such as cytokines or other growth factors, are then secreted by the cells and can be captured by that specific antibody on the surface of the ELISpot plate. After an appropriate incubation time, cells are removed, and the secreted molecule is detected using a detection antibody in a similar procedure to that of what you would see in an ELISA. The detection antibody can be either biotinylated and followed by a streptavidin enzyme conjugate, or the antibody can be directly conjugated to an enzyme. By using a substrate with a precipitating rather than a soluble product, the end result of this is visible spots on the membrane surface within the well. Each spot corresponds to individual cytokine-secreting cells, and the ELISpot readers, rather than being a colorimetric detector that you would normally see in ELISA, are actually camera-based and can identify and count the number of spots per well, giving you a quantitative measure. Simoa is developed by a company called Quanterix and stands for Single Molecule Array. And they have developed an approach for detecting thousands of single protein molecules simultaneously. Using the same reagents as a conventional ELISA, this method has been used to measure proteins in a variety of different matrices, including serum, plasma, cerebrospinal fluid, urine, cell extracts, and several others at femtomolar concentrations, offering roughly a 1,000-fold improvement in sensitivity. The approach makes use of arrays of femtoliter-sized reaction chambers, which are termed single molecule arrays, given rise to the name Simoa. And they can isolate and detect single enzyme molecules. Because the array volumes are approximately 2 billion times smaller than a conventional ELISA well, a rapid buildup of fluorescent product is generated if a labeled protein is present. With diffusion irrelevant at this size, the high local concentration of product can be observed, and only a single molecule is needed to reach the detection limit. Electrochemiluminescence is a kind of luminescence that is produced during electrochemical reactions in solution. And this particular technology is primarily the product of mesoscale discovery. The assay setup in an ECL assay is very similar to that of an ELISA, where you would coat an antigen down on a 96 or 384 well plate and apply your sample. The difference with this method is instead of a fluorescent or chromogenic detection, the secondary antibody used in ECL assays is conjugated to ruthenium, and the plates provided by MSD have electrodes on the bottom. Instead of adding a TMB or an ABTS, in order to read an MSD assay, you would add an aprotic solvent, and in this case, MSD's read buffer containing tripropylamine. So ruthenium, in the presence of an aprotic solvent, with the application of a voltage, produces light. So the basic setup is very similar to an ELISA, but the output signal is luminescent rather than chromogenic or fluorescent. So now that we've gone over a few of the assay formats of ELISA and those that derived from those basic principles, we'll touch on automation. And first of all, what does that mean? Automation, to some people, means large systems that process hundreds of plates in a day in a walk-away fashion. To other people, automation can mean simply using a liquid handler or a stacker. Me personally, I think it encompasses all of these things. Automating a basic assay format is not something that should be overlooked. It's not only complex assay formats that can benefit from a degree of automation. Now, so what does this mean? Are we just going to bring in a liquid handler or a dispenser? Automating an ELISA can be as simple as have a washer and a reader with a plate stacker. It's allowing the scientist time to do non-routine tasks while the routine work is being done by the equipment in the lab. So if you're going to consider automating a basic assay format, you need to decide what you mean by that. What do you hope to accomplish? Do you want to increase throughput? Do you want to standardize your sample handling? Do you want to free up scientists to do other things? And you also need to determine the scale and scope. How big do you want it, and what do you want it to do? There are some benefits and some takeaways to automating basic assay formats such as ELISA. With an ELISA, there is a lower bar of entry. You don't need to be an automation expert in order to successfully put an ELISA or an ELISA-like assay format onto some kind of an automation. One of the greatest benefits of automation is equal time treatment of samples. And we'll get into that a little later when we talk about the scheduling software. I tend to refer to automation assets within a lab as a force multiplier. You're allowing one scientist to do the work of two. If they can set up a platform and let it run while doing other things. It also provides a degree of consistency in your sample handling and your reagent dispensing. We all know how to pipette, and we should all know that we do not do it exactly the same time every time. There is a degree of variance with that. If you use a liquid handler, whether it's something like a multi-drop dispenser or a multi-channel liquid handler, setting up the same protocol and running the same method is going to be handled exactly the same way every time. Some of the liabilities when it comes to automating some of these types of assays is automation generally is not faster. It's take a little longer to complete the same task than it would doing it manually. There is obviously a cost involved with equipment purchase. Getting buy-in from management is another stumbling block for some. And there's also, the perceived lack of expertise. Well, this individual expert, but all these things can be overcome. As I said, some of the considerations you need to look at when thinking about automating any type of assay work within a lab is the time it takes. Assay transfer is something that cannot be overlooked either. Assay development is usually done it is a necessity to take your manual bench method, apply it to whatever automation you're considering implementing, and making sure that you get the same results. Training is something that gets overlooked more than people might think. Familiarity with the equipment and the software that you're going to be working with is extremely important when considering an investment like this and be provided to all individuals who will be using this equipment. Maintenance requirements are also something that a lot of people overlook. Service contracts have a cost. They also have a significant benefit, especially if you have critical equipment. Running a screening lab, we have several pieces of equipment here that we would consider critical, and we have service contracts on them that provide us 24-hour response time. Because if those pieces of equipment go down for any length of time, our workflow stops. Some individuals prefer to not have service contracts on anything. I've had managers in the past that have thought it was a waste of money. But different tiers, vendors will provide several tiers of service contract, and it's just a question of, if you decide you want one, finding what level of attention and cost suit your needs. Selection of the platform is obviously very important, and this is where loyalty to a particular brand or vendor is not going to serve you well if their instrumentation doesn't have the capabilities that you need. Some people know one type of instrument, one type of or one type of plate reader, and that's the one they want to stick with because they're familiar with it. But sometimes there are better options out there for your specific needs that need to be considered. Cost of consumables. When running automated platforms or automated systems, you're generally going to need an overage volume when it comes to reagents. You're going to be working out of larger reservoirs, so that needs to be taken into consideration. For ELISA assays, most of the reagents are generally not that expensive, so a little bit of overage isn't really that big of a concern. If you're running something like AlphaScreen by Perkin Elmer, those reagents are exceptionally expensive. So if you're going to be running that type of an assay on an automated setting, reagent overage is a significant cost consideration. And by comparison, so MSD, the reagents are relatively inexpensive. The plates, however, are considerably expensive given to the unique nature and the embedded electrodes. So all of these things need to be taken into account when proposing automating specific assay types. In touching back on the assay transfer, one thing that really needs to be looked at is that small changes in how you perform your assay can have big effects in an automated environment in resource allocation and time to completion. So at this point, I will go over our process here at Boehringer briefly. Obviously, I cannot provide a lot of detail as to what our discovery process is, but I can say that we do perform a large number of tasks, and it is a rather lengthy process, and we implement some degree of automation at several points throughout that process. All of those steps in the swim diagram here that you see that are in red boxes are steps in which we have some degree of automation, and that we have walk-away platforms, several workstations, or just a liquid handler to help with bulk transfers. As an example, I'm going to focus on the implementation of an automated platform for one of our primary screening methodologies that takes place in the beginning of our process, and that is MSD. So the reason that we decided to create an automation platform around the MSD was several-fold. We had the potential of running a high volume of plates, up to 120 plates per screen, in 384-well format. The assay method that we have developed here is not overly complex. It's actually relatively simpler than other methods. But there is a high degree of quote-unquote hands-on time required, which leaves the scientist that is running that particular screen unable to address other tasks. But given the high number of plates, it was very difficult to ensure equal time treatment between samples. Most of the components that went into the design of this platform we already had on hand from a previous platform. And as I've said previously, a walk-away automation platform such as this allows scientists to focus on non-routine experiments, assay development for other projects, that sort of thing. And we've already talked about the basics of the MSD assay format and how their technology works. It does have a significantly greater sensitivity than your standard ELISA. and can easily get down to picogram per mil, even femtogram per mil levels of some analytes. Given the way the detection apparatus works, is a CCD camera, and it only registers a luminescent signal within a certain distance from the bottom of the well. So as a result, any unbound conjugate in the fluid column that would normally contribute to background in other assays does not in this setup. So you have a very, very clean background signal in an MSD setup. We use two specific assay types here, one of which is an indirect ELISA, and the other is a hybrid indirect ELISA that we've developed in-house here. So our hybrid format is combining sample, our secondary detection antibody, and a biotinolated antigen, letting those reagents equilibrate overnight in polypropylene plates to reduce any binding to the plastic. The following day, that combined sample and detection reagent are applied to blocked streptavidin-coated plates, a 1-hour incubation, a standard wash protocol, add the read buffer, and read on the MSD instrument. Ours is a Sector S600. We also use an indirect ELISA standard type format when we have an antigen that cannot tolerate being biotinolated. So we will coat that antigen down on MSD's high bind plates, which are similar to any ELISA plate that you would see, like an Imulon or a Maxisorb. Add sample, incubate, wash. Add secondary detection, which is also the ruthenium labeled detection antibody, same detection antibody. Incubate, wash, add read buffer, and read on the MSD instrument. So we've developed automated methods on this new platform for both of these assay formats. The components of this Skynet platform, we have a Biomech FXP dual arm hybrid. So it's a 384 multi-channel head and a span 8 inside of a HEPA enclosure. Lyconic LPX220 hotel, ambient hotel. Biotech EL406 washer, plate washer, multi-drop combi, the MSD Sector S600 reader. We have a BERT2 extended range plate handler and two 39-inch servo shuttles. And this is the overall layout of our platform. And the only pieces of equipment for this layout that we had to purchase specifically for this are the ones that are highlighted in red. It is the BERT2 and the rear servo shuttle. So this was a bit of a budgetary experiment from our side of things. We wanted to build a platform to automate our MSD primary screening in a walk-away fashion, while also making use of components that were left over from a previous screening platform that has been discontinued to make the most of the equipment that we had. As I said, it was composed almost entirely of existing components, with only two needing to be purchased new. We also utilized Site Fabrication Shop, which does, amongst other things, metalworking, to create an enclosure on the Lyconic Hotel, which is featured in the foreground of the picture here. Since MSD assays are light sensitive and incubation is going to be taking place inside this hotel, we needed with an access door to go on the hotel. And our fabrication shop here on site at BI was able to do this for us at almost no cost. The total cost to implement this particular platform was less than $150,000. And if anyone has any experience with implementing setups like this, you will know that that is almost an absurdly low price tag for something like this. If all of the components were purchased new, with the engineering cost, this would easily be over $500,000. So I have provided some photos of our current setup. In the top left and top right picture is taken from behind the Biomech FX. So we have a servo shuttle that's actually coming out of the rear of the Biomech, and that's where the remainder of the components are in being serviced by the BERT, with the MSD reader, the multi-drop, and the plate washer. The Lyconic Hotel is connected to the Biomech by the second 39-degree servo shuttle, which you can see in the bottom left picture. And you can see the exit out the back of the Biomech in the bottom right. It is not, we'll say, the most elegant design, but it is definitely functional and really met our desire to do this almost entirely with pre-existing components. We do have, as you can see, an integrated barcode reader on the upper left photo, and in the bottom center, easy access into the MSD. And just as a side note, the MSD reader and any of the components on this system can still be used in an offline fashion. It can be used as individual instruments when the system is not running. So if someone needs to just run an MSD plate, they don't have to feed it into this platform. They can just use the MSD reader as a standalone. And Beckman Coulter was the integrator for this project. And what I mean by that, for those that are unfamiliar, the integrator is the vendor that will provide the scheduling software and do all of the engineering to make sure that all of the components are able to seamlessly communicate with one another. And why we chose Beckman Coulter as an integrator, if you have any experience with this type of equipment, you know, there's a lot of choices out there for integrators, and some of the best known are companies like Hamilton, TCAN, Hi-Res, Thermo, and even Perkin Elmer. The individuals that I work with here in my lab, we already have a large amount of expertise with the Biomech platforms, in particular, and other Beckman products in general. There were budgetary considerations around this particular platform that led us to stay with Beckman as the integrator for this platform, as they were the integrator for the previous incarnation of this platform. We do have individuals in our lab that have little to no experience with liquid handlers. So we do have a wide range of expertise. And personally, I have found that the Biomech software is the easiest to teach novice level users of liquid handlers. And also SAMI, Beckman's integrated scheduler software, is much easier to teach than some other scheduling softwares out there. And that's particularly my comfort level as the one that was going to be doing the majority of the training. We do have a lengthy existing relationship with Beckman Coulter, and we have close to a dozen liquid handlers within our department. So we do have a very solid relationship with Beckman as a company. And lastly, the quality of the service organization is something that we at BI place a lot of value on. We do have a long-standing relationship with our individual service engineers that are working on the majority of our platforms here. And it is something that we are very comfortable with. And all of these reasons together are why we chose Beckman Coulter as the integrator for this particular project here. And as I've alluded to a few times, SAMI is the scheduling software that Beckman Coulter uses for their integrations. So scheduling software is probably best described as an overlay for all of the components within an integrated platform. It's a controller software. It is the console that you will use to build your methods and control all of the individual components within your platform. And SAMI has a very easy to understand graphic user interface, which can show you visually how your protocol is going to progress, what labware is being drawn in to what step, where it's coming from, how long each step is going to take. Methods like this are built based on one plate. Build your assay format in this graphic representation about how one plate would progress through. A scheduler software will then take all of those processes and interleave them with one another to get maximum efficiency out of the process. As I said earlier, SAMI I have found to be one of the easiest scheduling softwares to teach new users. It also has an ability to stack protocols into a run queue, providing that your system has enough storage capacity and resource to handle both or 2 or 3 protocols in a row. If you can put enough consumables and plates and whatnot in there to handle everything that you need, it's a very easy function for stacking those protocols into a run queue, much easier than I found with other platforms. Back to something that I mentioned in one of the earlier slides, small changes leading to big impact. When I was doing the initial method development for this MSD screening assay, step approach that I used at the bench, and I put it into SAMI as I was doing it manually. The result was for a standard 30-plate screen would take over 14.5 hours, which is something that it was not viable for what we're doing. The simple action of swapping 2 steps in the protocol, it was basically just order of reagent addition, which is irrelevant to this assay setup, brought 14.5 hour time frame down to 5.5 hours, which was much more viable for what we were doing. And a lot of that is around resource allocation within an integrated system. If you have your liquid handler being used too much to move components around your system, that resource is not available to do any liquid handling. So that is going to greatly stretch out the overall time that it's going to take for something like this. So as I said, just swapping 2 steps within the protocol, it didn't fundamentally change the protocol at all. I just added reagent 2 first instead of second. And that brought the time down by over 70%. Resource allocation within an integrated system is the overall driver for the length of your method. And another thing that I had touched on in the preceding slides was equal time treatment. And what I mean by that is treating your samples in the exact same manner for every single plate. And that applies to liquid handling, mixing, shaking, washing, incubating, and reading. For example, our MSD reader, the Sector S600, for the 384-well plates that we use in our screens, has a read time of approximately 2 minutes per plate. So if I've applied read buffer to my plates, plate 1 goes right in, the wait time is negligible. Plate 30, at the end of that deck, has been waiting an additional 60 minutes. in the concept of a screening environment, those two plates are not directly comparable because one incubated for 1 hour, the other one incubated for 2. Setting things up in a scheduled environment, and you'll see some graphical representations here on the right side of the slide, are staggered over time so that each step has a defined time limit. And that's part of the assay setup. Your scheduler, through a variety of methods, you are going to tell it, or it is going to determine exactly how long each step within your protocol takes. And that's liquid handling, plate movement, washing, everything is going to have an execution time associated with it. That scheduling software will then put all of those times together and interleave all of these processes, so each step can occur with constraints that you have around it. So if you put into your protocol that you want your plate to incubate for 1 hour plus or minus 5 minutes, your software is going to schedule all the other tasks to make sure that that happens. And you'll get a telltale slanting of your process bars. And anyone that's ever worked in a scheduling environment knows exactly what I mean by this. In that way, you can directly compare plate 1 with plate 30. And since these are the same methods being performed over time, you can compare, plate 1 within your campaign to plate 400 in your campaign that was screened 4 months later, because they are directly comparable under the same methods with equal time treatment across a large facet of time. Wet testing validation is something that definitely needs to be done when implementing any kind of an assay transfer onto an automated platform. And what this means is you're just looking to make sure that all of your transfers are being done in the same fashion. And this also goes hand in hand with QC validation of any liquid handler that you have. And there's several services and technologies out there that can assist with that. But as an example here, I did a wet test validation of this MSD platform, and I ran a mock screen with 10 plates to assess reproducibility. The first and second column were background samples. The rest of the plate was a single concentration of a reference antibody. We also did this for the span 8, which was going to be dispensing our reference antibody control curves on each of the screening plates. And when we did this, we noticed that the first and second replicate of the reference antibody, the curves came in noticeably lower than the rest of the replicates. And the reason for that was after priming the span 8, we found that the weight that occurred from the initiation of the protocol to the first application of the control antibody, another priming step would have been helpful. So simply instituting a pre-wet step before the antibody was dispensed to the plate completely resolved that drift, and all of those curves are right on top of each other. But that's, it's small things like this. As I said, small changes can have big effects. And it's small things like this that need to be looked at. In conclusion, an assay is routine or basic doesn't mean that it wouldn't benefit from automating the entire or even just a portion of that assay. Consistency within single experiments and over extended periods of time is one of the greatest benefits for instituting automation of different assay types. As I've said, equal time treatment can allow you to directly and safely compare samples across a wide range of time. Wet testing validation of an assay is essential and critical. When looking at how to implement any type of automation or workstation approach, I always advise people to keep an eye to the future and make sure that whatever that they implement is scalable to future needs and also comes in line with your budgetary restrictions. I would like to thank LabRoots, and I would also like to thank Beckman Coulter for bringing me in to be a part of this educational webinar series. We have the necessary disclosures and copyrights that the lawyers have insisted, and I am free to take any questions.
Philip, thank you for your presentation. A quick reminder to our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Philip will answer as many questions as time permits. Okay, Philip, our first question. How do you minimize edge effect in your automated ELISA?
Well, there's several ways that you can go about minimizing edge effects. The easiest way to look at that is making sure that your plates are lidded during their incubation times. I would keep incubation times themselves as short as possible while ensuring maximum performance within your assay. Other things that generally escape notice are your lab environment in general. A high degree of air conditioning can cause very dry air, which can increase evaporation. Looking at the assay volume versus the plate type that you're working with. For example, if you're using a 96 well plate and doing a 50 microliter ELISA, small degree of evaporation is going to have a much larger effect than, say, if you were doing 100 microliter ELISA or doing it with a 384 well plate. Much smaller available surface area for that evaporation to take place. Another way of looking at it is incubating your plates within a humidified environment to keep that evaporation to a minimum. A lot of it depends on the resources that you have available and the general environment that you're working in.
Philip, our next question is two parts. Now, based on your experience, how easy was it to program an ELISA on the Biomech? And what advice, recommendations do you have for an automation novice?
In my personal experience, it's actually quite easy to program an ELISA protocol onto a Biomech. Assuming that you're working with a Biomech as a standalone liquid handler, it's basically just a series of single reagent transfer steps. And those can be done basically the same protocol with just a different source reagent. And that's probably one of the easiest methods to set up within the Biomech software. That gets obviously a little bit more complicated if you start integrating components on deck within a Biomech or any other liquid handler is part of an integrated system. When it comes to setting up liquid handling protocols around some of these assays, the best advice I can give, especially for novice users, is don't overthink it. If the thing you need to do is add a reagent to a plate, it's a simple single reagent transfer. Making a method overly complex is not going to benefit you in the long run. That obviously changes once you get into scheduled environments. The level of complexity increases when you're talking about integrated devices in a scheduled environment. And then we'll say within SAMI, as an example of a scheduled environment, setting up an ELISA protocol is also quite simple. The most part comes when optimizing that method for an optimal timing, which is part of what I touched on towards the end of the presentation there.
Thank you, Philip. And a reminder to our audience, if you'd like to submit any questions, there is a drop-down box located on the far left of your presentation window labeled Ask a Question, and just type in your question and click Send. Okay, our next question, Philip. What steps do you take to minimize sample-to-sample contamination during automation?
Well, there's multiple ways that you can look at this, and it also depends on what types of samples that you're referring to. Some people, in, we'll say, an effort to reduce consumables costs, institute tip wash. And most liquid handling platforms, Biomech included, usually come with the available option of a tip wash station. I would recommend a very hard evaluation, a very stringent evaluation of your tip wash protocol. Also, using new tips for every sample is an easy way to avoid cross-contamination. Using barrier tips is another way if you're working in more of a molecular biology standpoint or a phage standpoint. Barrier tips would be a necessity. I work a lot with cell culture supernatants and hybridoma cultures. So we have HEPA enclosures around several of our liquid handlers to reduce broad contamination overall. Like I said, it depends a lot on the types of samples that you're working with and the types of contamination or cross-contamination that you're worried about, but there's usually multiple ways to approach those problems.
Thank you, Philip. Here's another question. How do you go about troubleshooting an automated assay?
Oh, very good question. This is a primary reason why benchtop to automation transfer is so vital. If you develop your assay method at the bench manually, and you know that your assay is performing as expected, moving that over to a liquid handling platform or an integrated platform, you start to see discrepancies or drift. It's not the chemistry or the biology of the assay that's your problem. The best way that I can think to go about something like that is, and it does take a bit of a familiarity with the components that you're working with, looking at your point and identifying the possible causes, and then changing one variable at a time across your process to identify what part of that automation process is your culprit, so to speak. It does have to be a very methodical approach, and that is a big reason why I never recommend developing methodologies on automation platforms. I would highly recommend developing them manually at the bench and then transferring to an automated platform. It takes a whole set of variables off the table if you do start to see problems.
Thank you, Philip. And it looks like we have time for one more question. But I do want to remind our audience that any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Okay, Philip, our last question. How do you access and visualize data from your automated assays?
Well, there's a couple of things that come into consideration with that. Personally, the softwares that we use here at BI in my lab here are primarily GraphPad Prism and Spotfire for doing data visualizations. Accessing them is where you can run into some problems sometimes. Network connectivity is a big issue, and it tends to be a little easier in academia versus corporate environments. For example, our two automated platforms that we have in my lab now, one that was integrated by Perkin Elmer and one that was integrated by Beckman Coulter, neither of them are actually compatible with our corporate firewall. So those platforms actually are not on our network. So we have to physically transfer those files through a gateway computer. Those restrictions tend to vary, I guess we'll say, from company to company and academic institution to academic institution. But as far as the actual data processing and data visualization, the ones that we use are Prism and Spotfire, and I'm sure that there's going to be a preference with that with most users.
Thank you again, Philip Gorman, and thank you to our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand through August 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.